So insulin is a polypeptide hormone. When you say polypeptide, that is a polymer of amino acids that is joined together by peptide bonds. So it is produced by the beta cells of the islets of Langerhans in the pancreas, and it has a profound effect on carbohydrate, fat, and protein metabolism. For the structure of insulin, for the human insulin, it contains about 51 amino acids, and they are arranged into polypeptide chains. So we have the A chain and the B chain. The A chain is composed of 21 amino acids, and the B chain is composed of 30 amino acids, now for a total of 51 amino acids. There are two, so the link between the A and the B chain is made by the disulfide bond or the disulfide bridges. And there are two interchain disulfide bridges. We have the, and this is located between the amino acid number 7 of the A chain and the amino acid number 7 of the B chain. The other disulfide bridge or the interchain disulfide bridge is located between the amino acid of the number 20 of the A chain and the amino acid number 19 of the B chain. So when we say interchain, interchain disulfide bridge, this is the connection or the bond between that is located between the A chain and the B chain. So it connects the two chains at this particular location. We also have the intrachain disulfide link. So when we say intrachain disulfide link, that is a link that is located within the chain. So in insulin, the intrachain disulfide link is located in the A chain. And this is particularly located between the amino acid number 6 and amino acid number 11. So we have here the structure of the human insulin. So this is considered to be the primary structure of the human insulin. And this was drafted by Harkness and Craig in the year 1952. So again, insulin is composed of two chains. Now we have the A chain here that is composed of 21 amino acids, and the B chain here that is composed of 30 amino acids. So the A chain and the B chain is composed of amino acids now that is numbered from 1 to 21. The portion here is the N termini or the N terminal of our insulin, and here is the C terminal. So this is usually fixed for our this is usually a fixed amino acid for different animal species. So for example, here in human insulin, the N termini of the A chain is the glycine residue or the glycine amino acid, while its C termini is the asparagine amino acid. For the B chain, the N termini of our B chain is usually the phenylalanine, while its C termini in humans is the threonine, threonine amino acid. This is usually fixed in different animal species. And we also have here, as we have said earlier, the two interchain link. When we say interchain link, that connects the two chains of our insulin structure. So for example here, the A chain and the B chain is linked through cysteine residues at this particular location, and another link is located between these cysteine residues that is located near the C termini. We also have here the intrachain link, and that is located on the A chain. And for this example, the intrachain link is on the cysteine residues at the amino acid number 6 and amino acid number 11. We also have here a structure of another representation of the structure of insulin. So we have here the A chain and the B chain of insulin. So these are polypeptide chains that are connected through peptide bonds. And the two chains are connected by way of the disulfide bonds. So the A and the B chain, now for example, are connected at this level at the cysteine residues of the 7th amino acid of the A chain and the 7th amino acid of the B chain. Another link is located between the 19th amino acid of the A chain and the 20th amino acid of the B chain. And these are known as disulfide bond or disulfide bridges. We also have here an additional link that is known as the intrachain link. No, intrachain link, and that is located between the cysteine residues of the 6th and the 11th amino acid. So the A chain here and the B chain are numbered from the N to the C terminal. So for example here, the glycine here would represent the first residue or the first amino acid, and we number it from 1, 2, 3 until 21 at the asparagine level. So that is the amino acid that is located at the C termini. For the V chain, we have here phenylalanine, and that is numbered, the polypeptide chain is numbered from 1 to 30 until we reach here the threonine amino acid at the C termini. We also have here another representation, another example of the hormone insulin, particularly in the cow. So in the cow insulin, it is basically the same, almost the same to that of the human insulin. It is still it does contain it still contains 51 amino acid. So the A chain has 21 and the B chain has 30 amino acids. And in this type of insulin, again we have here our glycine at this terminal here or termini here, or the N termini of our A chain, and the asparagine at the C termini. For the B chain, we have here phenylalanine at the N terminal and the alanine at the C terminal. They are still connected by way of the disulfide bond at the cysteine residues at these two locations, and we have here an extra disulfide bridge at the 1, 2, 3, 4, 5, 6 and the 11th residue, cysteine residue. We also have here another representation of the primary structure of the insulin, particularly in the pig insulin. So this diagram here represents the primary structure of the pig insulin. This is a protein hormone as discovered by Frederick Sanger. So the same, we still have here 21 amino acids for the A chain and 30 amino acids for the B chain. So the C terminal here is still the same, rather N terminal amino acid here is still the same. We have here glycine and asparagine as our 21st amino acid. For the B chain, we have phenylalanine and threonine at the 30th amino acid. They are still linked together as the cysteine residues, particularly at the 6th and the 11th amino acid, and that is our intrachain link. For our interchain link, they are still here, cysteine residues, and there is also here a connection between the 20th and the 19th cysteine residues of the A and the B chain respectively. So insulins from different animal species contain two polypeptide chains and have high homogeneity and similar 3D structure. So insulins from different animal species show a residue activity or identity at the 22 amino acid position. So although they are said to have high homogeneity, there are still great variation, particularly variation that is located in the amino acid number 5 of the A chain, amino acid number 6 of the A chain, A10, amino acid number 30 of the B chain. But the biological function is still identical. So we have here a diagram now showing the variation in the positions in insulins from some animal species. So we have here a comparison between the insulin of humans, pigs, dogs, rabbit, cow, sheep, horse, and the sperm whale. So for humans, we have here variations in positions in insulin from different animal species. So as you can see here, for human, pig, and dog insulins, they are almost the same, in the sequence of their amino acid, but they only differ at the amino acid at the 30th amino acid of the B chain. So for example, between humans and pigs, they are almost the same, but they only differ in their 30th amino acid, located at the C termini of the B chain. So example for humans, the B30 is threonine. For pigs, that is alanine. For the amino acid or the insulins of pigs and dogs are almost the same, because they don't differ in this uh, diagram. So the same is true for rabbit. So for rabbit, they are almost the same, to that of dog, pigs, and humans. But it only differs on the B30. B30 uh, residue or amino acid. We also have here another example of the different animal-based insulins. And we also have here the man-made, or the synthetic type of insulin. So we have here the human, bovine, porcine, canine, and feline insulins. So they are compared according to their variation in the amino acid composition that is located at the A8. So when we say A8, that is an amino acid that is located at the 8th position of the A chain. When we say B3, that is the amino acid that is located at the 3rd portion of the B chain, the 3rd amino acid of the B chain. So when we are going to compare these animal species, for example, in humans insulin are almost the same to that of pigs. Now they are almost the same to that of pigs, but it only differs on the B30 amino acid. For the canines, or for dogs, so we can see here that it is still very the same in amino acid sequence to that of the pigs and humans, but it only differs on the amino acid sequence that is located at the 30th position of the B chain. For cats, it is different. The insulins of cats is slightly different from that of dogs. We also have here the different synthetic or man-made insulin preparations that are used for the treatment of diabetes. So for example, here we have Lispro, Aspart, and Glargine. So the animal-based insulins are useful in treating DM or diabetes mellitus in humans because all mammalian insulin is structurally similar and is composed of 51 amino acids that is linked by 2 polypeptide chains. So porcine insulin is very similar to human insulin and identical to canine insulin. So bovine insulin is most similar to feline insulin. So these three here, the human, porcine, and canine insulin are almost the same, but they only differ at the B30 amino acid. For those of the cattle insulin or bovine insulin is almost the same to that of the feline insulin, but it only differs in this particular amino acid. So although the amino acid sequence varies among species, certain segments are highly conserved, including the positions of the three disulfide bonds, both ends of the A chain, and the C-terminal residues of the B chain. So usually the ends, the ends of the A chain and the B chain are usually the same in most of the species, as well as the position of the 3-disulfide bond that links the two chains and the A chain. So these similarities lead to a 3D conformation that is very similar among different animal species. So insulin from one animal is very likely biologically active in other species. So for example, in the treatment of diabetes in humans, so porcine insulin can be used, or the insulin that comes from porcine can be used because it has almost the same 3D conformation and structure in terms of the AA sequence. So pig insulin has been widely used to treat human patients. So the biological activities of insulin are not highly species specific. So we have here similar amino acid structure that can be found for feline, bovine, canine, and porcine. But again, for the structure, in terms of their similarity, so for the cats or felines, they have almost the same structure to that of the bovine. For canine and porcine, they have almost the same chemical structure. So insulin molecules have a tendency to form dimers in solution due to hydrogen bonding between the C-termini of the B chain. So additionally, the presence of zinc ions, insulin dimers associate into hexamers. So these are the different structures of insulin when they are going to be subjected into a solution. So this is an insulin dimer, and this is an insulin hexamer. So the difference between the two is that for insulin hexamer, it has the presence of zinc ions. So for monomers and dimers, they readily diffuse into the blood, whereas hexamers diffuse poorly into the blood. So hence, absorption of insulin preparations containing a high proportion of hexamer is delayed and somewhat slow because of the presence of this. Again, because it is larger and because of the presence of the zinc ions, which are considered to be polar in nature. So the first of these molecules to be marketed is called the insulin lispro. So this insulin lispro is a man-made type of insulin. That is first molecule of insulin to be marketed. It is an engineered type of insulin such that lysine and proline residues on the C-terminal end of the B chain are reversed. So this reversal of the lysine and proline residue on the B chain, particularly near the C-terminal end, does not alter the receptor binding, but minimizes the tendency to form dimers and hexamers. So again, the monomers and the dimers are considered to be greatly absorbed. They are more absorbed compared to that of the hexamers. And that is why this insulin lispro was made in order to prevent the development of the insulin to form these dimers and hexamers. No, because again, no, the monomer one, the monomer, no, they are more absorbed in the body. So this is the structure of the human insulin as compared to that of the insulin lispro. So the insulin lispro have a reversed position of the lysine and the proline amino acid. And this is the C-terminus of the B chain. So this is an example of insulin lispro that can be used in uh, that has been used in veterinary medicine, particularly in dogs and cats. So this the brand name, no, of this insulin lispro injection is Humalog. So Humalog is a fast-acting type of insulin that is used to control blood sugar level or diabetes in dogs and cats. So this is important, no, as a supportive and ongoing treatment for the diabetes mellitus.